Okay, now we are going to continue with 6.3, okay, which is chromosomal mutation. Okay, for this chromosomal mutation, you have to know how to define chromosomal mutation, and you have to state two types of chromosomal mutation. Okay, the first one is change in chromosomal structure, okay, or chromosomal aberration, and the syndrome for this kind of chromosomal mutation is Cradusha. Okay, while change in chromosomal number or alternation of chromosome number. Okay, so there are two types of chromosomal mutation, change in structure and also change in number. Okay, so the syndrome for the chromosomal number or alternation of chromosome number is Down syndrome. Okay. Alright, C, you have to state the types of chromosomal aberration, okay, which consists of translocation, deletion, inversion and also duplication. Okay, while the types of alteration, we have aneuploidy, example of syndrome is Down syndrome, okay, and also euploidy, or okay, also called as polyploidy. Okay, so next is definition of chromosome mutation. Okay, chromosomal mutation means change in numbers or structure of chromosome. Okay, here we have human karyotype. Okay, as we know, human have 46 chromosomes, which have 23 pairs, and each pair consists of two chromosomes. Okay, so each pair consists of two chromosomes, but in this picture, okay, look at chromosome 21. Okay, here we have 3 chromosomes with one extra chromosome. Okay, so there is mutation occur, okay, which called as chromosomal mutation. Okay, and here also we have change in structure, okay, of chromosome. Here we have original chromosome. Here we have mutated chromosome, where the blue segment of chromosome is repeated. Okay, so next, okay, so we have two types of chromosomal mutation. The first one is chromosomal aberration, which change in structure, and chromosomal number alteration, which change in number. Okay, so for the chromosomal aberration, we have translocation, deletion, inversion and also duplication. While for alternation number of chromosome, okay, there is change in number. So we have euploidy, or called as polyploidy, and also we have aneuploidy. Okay, for polyploidy, okay, it involves the whole sets of chromosome. So the changes occur, okay, for the whole set of chromosome, while aneuploidy, okay, change in number occurs, okay, only involve individual number of chromosome. Okay, for polyploidy, we have two types. Okay, we have autopolyploidy and also allopolyploidy. Okay, alright, so we move on to chromosomal aberration. Okay, so the definition of chromosomal aberration is change in chromosomal structure. Okay, so you have to remember, aberration means change in structure which result in abnormalities in the structure of chromosome. Okay, this is due to errors in meiosis or mutation that cause breakage of chromosome. Okay, alright, so there are 4 types of chromosomal aberration. We have translocation, deletion, inversion and also duplication. Alright, so we move on to translocation first. Okay, so for the first one is translocation. Okay, translocation means that a segment of chromosome break off and reattach to another part of same chromosome or non-homologous chromosome. Okay, so the change in position of genes involved. Okay, there is no gain or loss of genetic materials. Okay, because it's only translocated. Okay, phenotype may change and become abnormal. Okay, so translocation divided into two types. So the first one we have non-reciprocal translocation and we have reciprocal translocation. Okay, let's compare these two. Okay, for non-reciprocal translocation, one chromosome translocate a segment to same chromosome or non-homologous chromosome. It translocate. But here the term is exchange. Okay, for reciprocal, exchange of segment between two non-homologous segment, and this occurs, okay, to the most common, okay, this is the most common types of translocation. Okay. Okay, back to non-reciprocal. Okay, for non-reciprocal, it divided into interchromosomal and also intrachromosomal translocation. Okay, for inter first. Intrachromosomal location, segment of one chromosome is translocated to non-homologous chromosome. It means that different chromosome. Okay, TER, different. Okay, for intrachromosome, okay, segment of one chromosome is translocated to another region of same chromosome. Okay, intra, same tra. Okay, A, intrachromosomal, same chromosome. Okay, and for reciprocal, as I said earlier, okay, there is exchange of segment between two non-homologous chromosome, and it is the most common type of translocation. Okay, next we move on to deletion. Okay, deletion is a segment where a segment of chromosome is removed or lost. Okay, so the affected chromosome missing certain genes and become shorter. Okay, so let's see the diagram here. So we have A, B, C, D, E, F, G, H, I, J. So after deletion occurs, segment C is deleted. So the chromosome become A, B, D, E, F, G, H, I, J, and it become shorter. Okay, next. Okay, for inversion. Okay, inversion is a segment of chromosome breaks off and rotate 100 degree. Okay, before reattach in the same chromosome, rearrange the linear gene sequence. Okay, this can alter phenotype because the expression of gene is influenced due to change in position of gene. So for inversion, it can alter phenotype. Okay, alright. So let's see the diagram here. We have, okay, a portion, a segment of GHI is inverted, so it become IHG. Okay, so it rotates about 100 degree. Okay, next. Okay, so type of inversion, we have two types of inversion. So the first one, we have paracentric inversion, and second one is we have pericentric inversion. Okay. Alright, for paracentric inversion, it does not include centromeres during mutation. Okay, so in this diagram, look at the centromere. So the inversion does not include centromere. So we have B, C, D portion. So inversion occurs, D will go up and B will go down. So after inversion occur, the sequence become A, D, C, B, E. Okay, but for pericentric, it occurs including centromeres during mutation. Okay, so here we have our centromere here. Okay, inversion will cross the centromere. So after inversion occurs, okay, so the portion of inversion involving D, E, F. So F will go up across the centromere. So it will become A, B, C, F, E, D. Okay, that one is for inversion. Okay, next, duplication. Okay, the definition of duplication is a segment of chromosome repeated. So the word here is repeated. Okay, result in the presence of repetitive gene in the same chromosome. Okay, so here we have normal chromosome. So the portion of DE is repeated one time. So here the portion of DE repeated for three times. Okay, so the chromosome become longer. Okay. Okay, now we are going to learn about, okay, alternation of chromosome number. Okay, so for definition of alteration of chromosome number, okay, so it is a change in chromosome number. So it involves chromosome number. Okay, so caused by non-disjunction of chromosome during meiosis, so leads to gamete formation with extra or loss of number of chromosome. Okay. So, non-disjunction. So, what is actually non-disjunction? So, non-disjunction is pairs of homologous chromosome fail to separate. Okay, the words here is fail to separate during anaphase 1 or sister chromatids fails to separate during anaphase 2 of meiosis. So, this occurs, non-disjunction occur during meiosis, either during anaphase 1 or anaphase 2. Okay, alright, so here we have non-disjunction. Okay, so let's look at this picture. Okay, so here we have non-disjunction during meiosis 1. For this diagram, okay, this occurs during meiosis 1. So non-disjunction occurs, so the gamete formation with extra one chromosome here. Okay, and this one with loss of one chromosome number. Okay, but for non-disjunction during meiosis 2, okay, this one we have one extra chromosome, here one loss of chromosome, but here we have a normal gamete produced, here also we have normal gamete produced. Okay, so non-disjunction, okay, so this non-disjunction occurs due to the failure of spindle fibers to form. Okay, so result in the production of gametes, okay, with abnormal, okay, abnormal number of chromosome. For example, we have n plus 1 or n minus 1. And for normal gametes is actually n, okay, not plus 1 or minus 1. Okay, so fertilization of abnormal gamete will produce abnormal zygote. For example, we have 2n plus 1 and 2n minus 1. Okay, alright, so the abnormalities is usually prevent embryo development or spontaneous abortion, or miscarriage can occur, or it will develop into individuals having abnormal characteristic. Okay, alright, so here we have a normal meiosis. Okay, meiotic diagram for normal meiosis. Okay, look at this picture. Here we have K2n is equal to 2. Okay, during meiosis 1 it occurs normally. So this cell receive one chromosome, this cell receive one chromosome. So n is equal to 1 because it's already divided. Okay, but okay, during meiosis 2, okay, it also occurs normally here. So after sister chromatids separate and move toward opposite poles, okay, each cells also receive one chromosome. Okay, so n is equal to 1, n is equal to 1. Okay, this also n is equal to 1 and n is equal to 1. Okay, but for non-disjunction, okay, so look at here first. Okay, non-disjunction during meiosis 1. Okay, so these two chromosome goes only to one side, okay, or goes only to one cell. So here this cell receive no chromosome. So at the end of meiosis, and at the end of meiosis 2, okay, this cell receive one extra chromosome, same goes to this one, but for these two cells it does not receive any chromosome. Okay, so n plus 1, n plus 1, n minus 1, n minus 1. Okay, so look at this picture. Okay, here we have non-disjunction during meiosis 2. Okay, look at meiosis 1. Okay, so this chromosome divide equally. So this cell receive one chromosome. This cell receive one chromosome. Okay, so for this cell, okay, sister chromatids separate and move toward opposite poles. So for these cells, okay, each cell receive one chromosome, so it will have a normal gamete. But for this one, okay, during meiosis 2, or specifically during anaphase 2, only these cells receive 2 chromosomes. Okay, so it become n plus 1. Okay, but these cells receive no chromosome, it become n minus 1. And okay, so if fertilization occur between normal gamete, which is n, and abnormal gamete, for example, we have n minus 1. So the zygote produced is 2n minus 1. Okay, so this condition known as monosomy. So here we have normal gamete and fertilized with n minus 1. So the zygote produced is 2n minus 1. Okay, if normal gamete, okay, fertilized with abnormal gamete, which is n plus 1, so it will produce 2n plus 1, which the condition is known as trisomy. Okay, so this one n plus 1, so it will have 2n plus 1 zygote. Okay, so